Hello, in this video we will discuss about the evolution of seeds. So the seed forming plant are spermatophytes, and we will also understand about the basic concepts and predominant our non-seed forming plants. So the spermatophyte is the predominant, and the basically seed forming plant are spermatophytes because sperma means seed. So the development of seed forming plant habit occurred approximately 390 million years ago, and but the complete seed forming plants appeared in late Devonian period about 360 million years ago. So in this way here is the definition. If we will discuss about the definition, so what is seed? Seed can be defined as fertilized ovule. So after the two gamete formation to become a diploid formation and contain integumented in indehiscent megasporangium. Indehiscent megasporangium integument is specialized and protective covering around megasporangium. So this is the basic concept, and on the other hand here is the step. Let's begin to understand for the evolution of seed. So the step one is the evolution of heterospory because there are beginning of the bryophyte and pteridophytes. It is the non-seeded vascular plants. It is non-seeded and are homosporous because the spores are homosporous, same spores, except Selaginella and some others. But in this case, you can see that it produces one kind of spores, and Selaginella produce two kinds of spores. So this is the first evolutionary process which that gives the heterospory. Hetero means different and spores means spore. So here is the sporangium which that contain male, and it is the homosporous ancestor which that is the same spores are present in the plant, while to become a heterospory. Heterospory contain microspore and megaspore. Microspore means the female spore and megaspore means the male spore identification. So in this way here is the microspore I am drawing here again for understanding as well. So this is the homosporous ancestor. After this homosporous ancestor will become a heterosporous ancestor. So this is the spores and this is the sporangium. This sporangium is basically the important part for the spore producing organ. So that is known as a sporangium. While the spore inside is the haploid or diploid, on the basis of the different species will produce the new plants. While here is the heterospory. Hetero means different. So the microspore and megaspore formation. So the megaspore is a precursor of the female gametophyte, while the microspore is the precursor of the male gametophyte. So this is the microspore and this is the megaspore. This is the megaspore. And in this way this is the heterospory formation. And after this it will be shedding and it will be moved into the soil after the maturation. After the maturation it will be moved into the soil and the male gametophyte will be developed separately while the female gametophyte will develop separately, and these two gametophyte will fertilize together to produce again two type of spores, megaspores and microspores. So this is the life cycle is known as the alternation of generation. During the evolution of heterospores, the alternation of generation, the gametophytic generation will be reduced while the sporophytic generation will enhance. For example, Pinus. So this is the basic concept. Let's begin to understand the retention and germination. After this basically is the shedding, but it want to retain, the plant want to retain inside the megaspore. Why? Because of water deficiency. So the bryophytes also known as amphibian plant because it requires the water for gamete fertilization, while the spore production requires the dry weather. So it's the retention and germination of megaspore within megasporangium. So the megasporangium is the part of the megaspore which that is retained inside, and in Selaginella the megaspore does not shed from megasporangium but megaspore develops into megagametophyte, sorry, inside the megasporangium, in megagametophyte in the egg develops. So here let's begin to understand this is the two type of sporangium, the microsporangium and megasporangium. This is the microsporangium and megasporangium is the male and female. And in this way here is the shedding of the microsporangium while the retention of the megasporangium. After this formation will the step 3 is the development of productive layer, protective layer around megasporangium. So the around megasporangium, the protective layer is known as the integument around the megasporangium. And on the other hand, the ovule formation. And ovule, what is ovule? Or we will discuss also. So this is the basic structure which that is showing the integument around megasporangium and inside the megaspore, while the ovule formation are present in the uh, and ovule is an integumented indehiscent megasporangium contain megasporophyte. Other words, ovule immature seed. So the ovule is the immature seed. After the evolutionary process will develop the mature seed and how it is possible. So remember again, the megasporangium is the female character while the microsporangium is the male character, which they are developed into the again pollen, and it is known as the anther, containing anther. So here is the ovule formation. And on the other hand, here is that you can see this the reproduction of a single functional megaspore per megasporangium is the major character. Why? Because the requirement of the nourishment and food and nutrition will be limited. So in this way, as well as the 4 megaspores formation in the female sporangium, which contain integument, will reduction to 1 megaspore and endospory formation. Endospory means the inside the spore will retain and will not burst the megasporangium for releasing. So it will be developed into the female gametophyte. And nucellus is here. Integument will be produced, and the sporangium enveloped by integument will produce and germinates the megagametophyte after the germination of the megagametophyte. So after these things, the development of embryo sac within megasporangium is very important. In this way, a single megaspore retained in megasporangium germinates to form megagametophytes or embryo sac that contain egg. So in this way, the embryo sac contain egg, and in this way here is the in sporangium. For example, basically in sporangium, development of embryo sac within megasporangium. So this is the 5th step, and in this way, remember about that a single megaspore retain in megasporangium and germinate to form megagametophytes or embryo sac that contain egg. So in this way here you can see this is the basically the germinates to form megagametophyte or embryo sac that contain egg. So you can see this is the female gametophyte which contain nucellus and integument, and in sporangium it is sporangium, and the 6th basically the next stage is basically embryo sac formation. In this way the embryo sac formation and the pollen will attach due to the development of the pollen tube, and in this way the modification of distal end will be occur. After the modification of the distal end will create the pollen tube for move the nuclei of the male nuclei inside of the embryo sac for fertilization with the egg. So after the egg will be fertilized here inside the egg, and after the sixth step is the modification of distal end of megasporangium for pollen capture. So the distal end will contain a sticky mucilaginous secretion, which that is the pollen male gamete spore will be intact, and after this intact to the pollen tube movement inside the pollen tube through is enzymatically, and integument is here and egg here, and it can be fertilized to produce the after the fertilization with the pollen with the egg to haploid to form a diploid, and this is the basically the gametophytic generation after the sporophytic generation. So the sporophytic generation will be reduced while the gametophytic generation will be reduced while the sporophytic generation will increase due to the dry condition. So here is the seed formation. In this way, the cotyledon, epicotyle, hypocotyle formation, and radicle is here, which that produce the roots, and epicotyle will produce shoot system, and cotyledon used for the nourishment of the seed, and the megasporangium contain megasporangium will be contain here the degenerate. So let's begin to understand the summary of the evolution of seed. So the evolution first is the evolution of heterospory, and the second is the retention, germination of megaspore within megasporangium, and after this the development of integument as a protective layer around megasporangium, and after this the reduction to a single functional megaspore per megasporangium. After this, basically the development of embryo sac within megasporangium will be formed. And development of embryo sac within megasporangium will lead to modification of distal end of megasporangium for pollen capture. So the reduction to a single functional megaspore uh, per megasporangium. After this development of embryo sac within the megasporangium and embryo sac formation. After the embryo sac formation to modification of distal end. Distal means the uh, upper part of the female reproductive system contain ovary. So end of megasporangium for pollen capture. So this is the evolution of seed which that develop, and there are different process during the evolutionary process. So next video we will understand about the double fertilization and how to produce seeds. Thanks.